Happy Friday!
Happy Friday!
Shall we all celebrate? Call off class? Don't quote me on that. How's everybody doing? Everything making sense?
So far.
Mostly? Mostly? Working through the problems? Recitations? Okay. Everybody's got a smile on their face. That's good. So happy to see that. Okay. So as always, if you have problems or questions or there's things that aren't working right, let me know. That's my job to obviously, hopefully help you to make things right. So that's what I want to do. What I want to do today is talk about amino acids, because we're going to start turning our attention now to protein structure a bit and getting a better understanding about how these things that we've been talking about with respect to pH affect charge and how charge can affect the structure of proteins. As I've alluded to in the very first lecture, structure is essential for function. And so things that affect structure, we really need to have an understanding of. Okay? So that's the topic of today's lecture, and will be the topic of actually the next 3 lectures, including today. So as you can gather by the fact that, here we go, okay, we'll try this. And yes, I have it on vibrate so that you guys can't hear it. But my vibrate seems to make that thing vibrate. Okay. Shutting down. By virtue of the fact that I give 3 lectures on this, it says something about the importance of the topic of protein structure. A very, very important thing for us to understand. Well, structure is a pretty remarkable thing, all right? We think of protein structure as actually occurring at 4 different levels, and I'm going to talk in some detail about each of those 4 levels. The 4 levels are what we call primary, secondary, tertiary, and quaternary. Okay? Primary, secondary, tertiary, quaternary. And those 4 levels of structure you can think of as resulting from interactions between amino acids that are farther and farther and farther away. So therefore, the interactions between amino acids that are the closest are those of primary structure. The next closest, secondary structure, the next closest, tertiary structure, and the farthest away, quaternary structure. And as I describe these, I think you'll see why that's the case. Okay? So I want you to keep those in mind. They're not difficult concepts. They're not difficult concepts at all, but because of these 4 levels of protein structure that exist, all of the proteins that exist in the world can be explained and understood. Okay? Structure implies function. This is one of the best examples I can give you of structure determining function. Next term in BB 451, I will talk about this protein. This protein is called pCNA. You don't need to know that for this. The important thing about this protein is it helps DNA polymerase to stay stuck to DNA. Okay? Stay stuck to DNA. DNA polymerase copies DNA. It's got to move along the DNA. DNA polymerases that bind to this protein remain stuck to DNA for a long time. Why? Well, the structure of this protein is a ring, and like a ring sliding along the DNA, it doesn't let anything go away. DNA polymerases that stick to this protein stick to the DNA. DNA polymerases that don't stick to this protein go along the DNA for a little ways, they pop off, they pop on, they pop off, they pop on. Okay? Meaning, therefore, structure is, again, implying function. If they don't have the ability to bind to this, this protein itself binds to DNA because of its ring structure. This ring structure is essential for the function not only of this protein, but the proteins that interact with it. Okay. I like to show this figure because it's a really nice example when we look at proteins of how what I like to describe nano machines can assemble. When we look at, for example, a virus is composed of a protein coat and a nucleic acid inside of it at its very simplest level. Okay? That protein coat is a protection for the amino acids that's inside. And that protein coat has to be assembled. Well, viruses don't have factories. They don't have workers that can hammer rivets and make things stay together. They don't have glue. Instead, they make proteins that do something really remarkable. Those proteins can self-assemble. Okay? Now imagine having a puzzle that you put together that's got 500 pieces, and the puzzle puts itself together. That's what proteins in a viral coat can do. And understanding how that assembly process works has medical implications, as we will talk about later. And it has tremendous implications for the virus being able to make copies of itself, because if it can't assemble its protein coat, it can't function as a virus. So when we think about what I like to describe as nanomachines, and we have a lot of people who are interested in nanotechnology, it doesn't get any more nano than this right here. Things that can put themselves together. Remarkable, remarkable proteins. All right? Another important concept that I'll talk about with respect to structure of proteins relates to it indirectly. And that is the concept that proteins are flexible. This is a very important concept. You're going to hear me talk about the flexibility of proteins over and over and over this term. Okay? Flexibility is key to understanding what proteins do. It's key to understanding what enzymes, which of course are also proteins, do. And this example here is a prime one. It shows the effect of binding of a single atom of iron to a protein. A single atom of iron. When this protein binds to this atom of iron right here, the shape of this protein changes from Pac-Man over here to, I don't know, globular nightmare, all right? But it changes its shape, all right? And it changes its shape, has very drastic effects for the function of this protein. This protein over here will do something that this very same protein over here will not do. These are the same protein, differing in shape by the binding of a single atom. Okay? We will see later when I talk about enzymes that this flexibility allows enzymes to do things that chemical catalysts cannot do. Okay? It's the key to why enzymes can speed reactions trillions, quadrillions of times. Flexibility. So flexibility is a very, very important concept for us to understand, and it's a very, very important phenomenon for a protein to exist. Well, as we talk about and think about proteins, we have to go down to the level of the building blocks, and of course everybody in basic biology, I hope, knows that the building blocks of proteins are amino acids, and there are in fact 20 amino acids that are most commonly found in proteins. There's another amino acid called the 21st amino acid that is occasionally placed into proteins. And of those 20 amino acids that we always find in proteins, some of them get chemically modified after they get in. So we see quite a variety of things arising from the structures of amino acids in proteins. There are some things that we see not so variable, however. All right? One of the almost invariant things that we see in proteins is that when we look at the stereoisomeric configurations that exist of proteins, what we discover is that there's a very strong bias for biologically made amino acids. We can synthesize amino acids in a test tube. If we synthesize amino acids in a test tube, we get a bias, we get a mixture. Okay? They have two stereoisomeric configurations. You can see the configurations on your screen, D and L. If we make them in a test tube, no cells involved. We make them chemically, we get 50% D, we get 50% L. If we examine the amino acids that a cell makes, if we examine the amino acids that are in proteins, 99.999% of them will be in the L configuration. Okay? There's only a very tiny number of amino acids, and I'm going to actually give you the exceptions later in the term, of amino acids that appear in proteins that are in the D configuration. How do cells make such a bias? Well, they make a bias by virtue of the fact that the enzymes that make the amino acids have their own bias. They will only make one type preferentially. Why do they only make one type? They only make one type because they can only use one type. Well, how about that cell over there? Well, that cell over there is using amino acids from proteins it's gotten somewhere else. The language of biology says that if it's not L, I can't use it. So if I'm a cell and I've got D amino acids, I couldn't use all the L's that were out there. I couldn't eat. So by default, everything ends up being L amino acids because that's what cells universally use. Now, this is a really cool phenomenon because if we are interested in a phenomenon of what's called astrobiology, is there life out there floating around out there in space? There probably is. Okay, we would like to know that. One of the things that people do when when meteorites fall to Earth is that they grab those, they try to get them before they get any contamination, before people get their hands on them, before all the Earth can get its contaminating amino acids in it. They bust open the meteorite and they analyze the amino acids that are in it. And yes, I keep saying asteroids. Meteorites are full of amino acids. The question then is, do we see a bias? Were those amino acids made like I make them in a test tube? Or were they made by an organism that has its own built-in bias for those structures? Okay? Well, so far we don't have any meteorites that show any strong bias that we can tell that. Okay? And we probably won't. Floating out in space is not a real good place for life to be. But it doesn't hurt to look, right? So it'd be kind of cool if we could see, we find a meteorite that has everything in the D or everything in the L, either way. It'd be kind of cool. We know that it didn't happen by that simple chemical process that we can do in a test tube. Gesundheit. Okay, now, I'm not going to ask you to draw, actually, let me go back, I'm jumping ahead. I'm not going to ask you to draw an amino acid in the D versus the L, okay? You're not going to have to do that. You should know that L's are the predominant form, and you should know the constituents of an amino acid. That's something I forgot to mention here, okay? Every amino acid has 4 basic constituents that we can point to, okay? Actually, we have 5 if we count the central carbon. There's a central carbon called the alpha carbon. Attached to that alpha carbon are 4 different things, and because there are 4 different things attached to it, that's why we have stereoisomeric forms of each of the amino acids, all but one, that is. Okay? 4 different things. One is called the alpha amine. It's this blue guy over here. An alpha amine, okay, is attached to an alpha carbon. We also have something called an alpha carboxyl. An alpha carboxyl is attached to an alpha carbon. In addition, we have a hydrogen. And the 4th thing that we have is an R group. Now, if you look at this, all the amino acids have a hydrogen. They all have an alpha carboxyl. They all have an alpha amine. The only way that the 20 amino acids differ from each other in structure is in the configuration of their R groups. So we can think of the R group as really defining the chemical properties of the amino acids. Okay? So if we understand the R groups, we understand the chemistry of each of the amino acids. All right. Well, I've been talking for a couple of days about ionization. I want to point out to you that ionization is critical for amino acids because amino acids have ionizable groups. Now, so far, the ionization that I've talked about has been about acetic acid. Acetic acid has a single ionizable group. It has a single pKa. All of the amino acids that we find in biology, all of the amino acids have at least 2 ionizable groups, and therefore at least 2 different pKas. At least 2 ionizable groups, at least 2 different pKas. The alpha carboxyl can ionize. It starts out as a COOH, which has a charge of 0. When it loses its proton, it has a charge of -1. The alpha amine can also ionize. Okay? If it has an NH2, which is what we see here, which is what we think of as an amine, it has a charge of 0. But if it gains a proton, it has a charge of plus 3. Plus 1. NH3 plus 1. Right. Okay? So two possibilities for each one. A charge of plus 1, a charge of 0 for the amine, a charge of 0, a charge of minus 1 for the carboxyl. Okay? The amine has its own pKa. The carboxyl has its own pKa. Let's examine what happens during the loss of protons with this amine. Let's say we start over here with all of the protons on. Okay? What would it take for me to put all the protons on? It would take a low pH, right? Today I'm going to give you a rule that I want you to understand. It's going to simplify things for you, and you can actually derive this rule mathematically, or you can memorize the rule. This is one rule I won't put on your exam. Okay? We are concerned in ionization about protons being on or off. Okay? We're concerned about them being on or off. The rule I'm going to give you is as follows. Okay? If the pH of a solution is 1 or more units below the pKa, the proton of that group is on. The pH of a group, pH of a solution is more than 1 unit below the pKa of a group, the proton is on that group. If the pH of a solution is more than 1 unit above the pKa of a group, the proton is off. And you're saying, what if we have it somewhere in the middle? Well, if we have it somewhere in the middle, some molecules will have it on, and some molecules will have it off. We can't say one versus the other. Okay? Now that's what this graph on the screen is showing you, and I'm going to describe it to you. Does anybody want me to repeat the rule? Okay? pH more than 1 unit below the pKa, proton on. pH 1 or more units above the pKa, proton off. Okay? 1 or more below, 1 or more above. Right? Okay. Well, here is a plot that shows us, it's a little confusing of a plot. We'll just first look at the things up at the top. You told me that we had to have a low pH to start if we had all the protons on. Well, we know we've got all the protons on because there's the proton on the carboxyl group. That carboxyl group has a charge of 0. And there's that extra proton on the amine group, and it has a charge of plus 1. What would it take, for me to pull a proton off of this molecule? An increase in pH. How much would I have to increase the pH to know that I got a proton off? 1 or more units above the pKa, and you say, well, which pKa? Well, I haven't given them to you. The pKa of an alpha carboxyl group is approximately 2.2. I'll give you that on an exam. Okay? The pKa of an alpha amine group is approximately 9.5, and I'll give you that on an exam. Okay? So if I wanted to pull off, so if I have this guy has a pKa of 2.2 and this guy has a pKa of 9.5, which proton is going to come off first? The carboxyl, because the one that's got the lowest pKa. Which one's the stronger acid, the carboxyl or the amine? The carboxyl, because it's got a lower pKa, right? Okay, basic rules. All right, so how high would I have to raise the pH to be pretty sure I've got the proton off of the carboxyl? 3.2, 1 or more units above, right? All right, so when I look out here at about 3.2, look what's happened. We've pretty much gotten this proton off, and that's what this graph is showing. The percentage of pink is dropping till we're essentially down here. We've got the proton off. Instead of having this molecule, we essentially have this molecule. It was until we start pulling it off. So we're pulling a proton off here. Excuse me. Pulling a proton off. I'm sorry? Because I'm getting above the pKa. That's right. So notice the pH is rising. Well, what if I was at 2.2? Well, if I was at 2.2, what would I have? I would have half and half, right? Half of this guy and half of this guy, right? Notice both these guys have the NH3 plus. Why is that? Because at 2.2, I'm more than 1 pH unit below the pKa of the amine group. Amine group proton stays on. Okay? If I said, which of these two is the salt and which of these two is the acid, you would say, the salt is? The salt is this guy right here. The salt will always have one less proton than the acid. This has the most protons. This has lost a proton. Salt's in the middle. There's the acid, right? That's going to change over here between these two. Which one's the salt and which one's the acid? Salt on the right, acid in the middle. So whether something is a salt or an acid depends upon which ionization we're talking about. Okay? Well, notice, we keep adding sodium hydroxide, we keep adding sodium hydroxide, the pH keeps changing, and the pH keeps changing, and all of a sudden we start seeing the green form start to appear. And where is the green form going to start to appear? Well, within about 1 pH unit, things a little bit more exaggerated than mine, but within about 1 pH unit of that pKa, we start seeing ionization happening. By more than 1 pH unit above the pKa, it's essentially all happened. At a pH of 9.5, you would expect that we would have approximately 50% this, 50% this, and that's exactly what we have. Okay? So this graph is showing you, okay, in graphic terms, what's happening with these molecules. Notice, and this always confuses people, there are 3 molecules. There are 2 ionizations. pKas refer to ionizations, not to molecules. Okay? How come there's 3 molecules? I've only got 2 pKas in this problem. Well, think back to what I just said. pKas refer to ionizations, not to molecules. They refer to the process of this happening, where this happens. Okay, questions about that? No dazed looks? You guys look like that's okay? Yeah? In this case, this molecule is the salt, and this molecule is the acid if we're talking about this one. And specifically, you're right, this guy is the thing that's that's lost the proton, this is the thing that's gained the proton. But specifically, it's the entire molecule. Okay? Okay, good. Oh, I thought I saw a hand. Okay, yeah.
So the third can only ever be an acid, and the first could only ever be.
This can never be an acid.
This can't give a salt.
This can never give up a proton. That's right. Yeah. Okay. And this could only ever be an acid. That's right. All right. Good. Very good. Let's move forward. Now, first of all, I'm not going to ask you to memorize the structures of the amino acids. Okay? If you're in the majors class, I would expect you to memorize all 20 structures, and you would really love me. But now, because I didn't make you memorize that, you really should love me, right? I need love. All right. But, and there's a big but, I said that the R groups determine the chemistry. You should know a bit about the R groups in terms of categories. All right? Now, your book in this edition went to something that's a little different scheme than most books use, and I'm going to use their convention to keep it simple for you and so you won't get confused. If you look in the 6th edition of the book, you're going to see this is going to look different. So you might want to refer to the figures of the 7th when you're learning your amino acids. Okay? Your book groups amino acids into several categories. One category you see on the screen are what they call hydrophobic. And they're called hydrophobic because they have R groups that will not interact with water very well. They don't like water very much. Okay? So if I ask you to identify the hydrophobic amino acids, I would expect that you would know this. You should know the names of all 20 amino acids, yes. But you should know when I say hydrophobic amino acid, you're going to have these things pop into your head. Oh, there's valine, there's leucine, there's proline, there's also other ones. And these include these guys here. These are all in the category of hydrophobic. They have side chains that really don't interact very well with water. Some of these have big side chains. Look at tryptophan. That's the biggest side chain right there. Okay, that's a big honking molecule, and it doesn't like water. Now, this hydrophobic nature of these side chains of these amino acids have very important implications for the location of amino acids in proteins. I'll talk about that later when I talk about tertiary structure, but I want you to keep that in mind. Okay? The chemical nature of the R group will determine a lot about where these things are found in proteins. Okay, another group that your book refers to are what are called polar amino acids. Okay? Polar amino acids have side chains that interact with water very well. They're either ionic or they have something that can hydrogen bond. Okay? Cysteine, for example, can ionize its sulfur, its sulfhydryl reasonably easily. It will interact with water very favorably. Threonine, hydroxyl side chain, hydroxyl group. When we think hydroxyl group, we think hydrogen bond, therefore likes water. Okay? So these are polar amino acids. They tend to be hydrophilic, liking water. Okay? They will hydrogen bond or ionize. We'll see there's a separate category that ionize, and I'll describe those to you here. But these guys here, if they ionize, it's usually not to a large extent, with the possible exception of cysteine. Cysteine actually ionizes reasonably easily. Okay. Now, here's a group that I call the positive, what happened there? Called the positive R groups, all right? I've got the wrong figure linked. Okay. Let's see. Oh, blast it. Okay, I'll have to fix that. This category includes lysine, arginine, and histidine. What you see on the screen here is only the ionization for histidine, unfortunately. I thought I had all three of them up there for you, so I'll fix that. But these guys all have side chains that have amine groups. They all have side chains that have amine groups, and therefore that means if they have a proton on them, they will be positively charged. So these guys can have an R group that definitely is positively charged. Now, the R groups of these guys vary, but if I said to you that the R groups of these amino acids side chains are on the order of 10 or 11, what would you say about their charge at physiological pH? Are they charged? Are they uncharged? Are they positive? Are they negative? What are they? pKa of, let's say, 10. Physiological pH is 7. The rule tells you what about the proton? Proton on, right? pH more than 1 unit below the pKa. We're talking about an amine group. Proton on an amine group. Charge? Positive 1, right? Okay, so this is the kind of thing you should be able to go through in your head just like that, just like I'm doing here. It's not hard, but when you get the basic rules, you'll understand these components of charge. Okay? Now, histidine actually is an exception. It has a rather odd pKa, but I won't talk about that at the moment. Okay. There are 2 amino acids that ionize very readily, okay, in their R groups at physiological pH. These are the negatively charged R groups. They are what we oftentimes refer to as the acidic R groups. By the way, the last group your book calls basic R groups. I tend not to like that term, but if you want to call it that, that's fine. Okay. I like it the positively charged R groups. That's the way I like to think of them. All right. These guys have carboxyls in their R group, and they have a pKa typically of about 4.4. Again, I'll give you that on an exam. You won't need to know that. And at physiological pH, if the pH is 7 and these guys have a pKa of 4.4, you should run through your head, well, the pH is more than 1 unit above the pKa. Proton will be off. Proton off the carboxyl, negatively charged. So these guys are usually negatively charged when they're found in proteins in cells, or when they're found in cells alone, either way. Okay. Now, you do not need to memorize the 3-letter abbreviation. You do not need to memorize the single-letter abbreviation. you do need to memorize the names. Okay? Do I have to spell them correctly? Well, since something like aspartate really isn't a very difficult word, I would say in general, yes. I'm not inflexible except for graduate students. Graduate students have to spell everything precisely. Okay? But we will not have a very wide latitude for something that's a simple name. I'll tell you that. Okay? Aspartate is aspartate. It's not asparamarl or bartlebate. And I've had students coming off. I've had students on an exam. Well, it was close, and I said, no. You need to know the name. So, but again, it's not absolute for undergrads, but it has to be pretty close. Okay. Here's a pKa table showing you some of what I've just described to you. And though I think their numbers in some cases are a little odd, I'll show it to you. Here's a terminal alpha carboxyl, okay? Approximately 3.1. Most of them are actually below that. Here's acidic amino acids, acidic side chains, about 4.1. I told you histidine was a little odd. Histidine's about 6, all right? And again, you don't need to memorize these at all. I will give you any relevant pKas that you need on an exam. But I just show you these to show you the various groups. One that's of interest is cysteine. I'll talk a lot about cysteine this term, okay? Cysteine ionizes reasonably readily, okay? 8.3. There's that stupid bouncing thing. Okay? And not only does it ionize readily, but it turns out that sulfhydryl, that SH group on the side chain of cysteine is very chemically reactive. It will readily react with other sulfhydryls of cysteine and make chemical bonds. And we'll see that this is an important consideration in stabilizing the structure of many proteins. Tyrosine has an OH that can go to an O-. It takes a fairly high pH to get that proton off, but it can happen. If I had pH 12, this tyrosine would have a charge like this. Okay? Lysine, of course, the positively charged polar side chain there. Arginine is even higher. Okay? Arginine, by the way, has a resonant structure, and we will just treat it as if it's a single NH3. Okay? We won't treat it as which one is which. It's resonant, and it's possible to go to either one. So we'll treat it as if it has a single NH2 that can become an NH3 out there. Okay. There's the abbreviations that you don't need to know. Okay. And I think we're there. All right. Now, this figure shows you very much what I showed you in that earlier figure. It's actually not even quite as nice as that first figure that I showed you, the ionization. This is a simple amino acid that has 2 ionizable groups. An example might be alanine. Alanine only has 2 groups that can ionize. The R group of alanine can't ionize. If I had aspartic acid up here, I would have 3 ionizations that could occur. Okay? Well, this actually comes up as important when we think about titration. So I showed you a titration curve the other day for acetic acid, or actually it was an acid I made up. It had a pKa of about 2.5. I showed at the end of class the other day. And you saw that single flattening. And that flattening corresponded to the buffering region. That was the place where that buffer was resisting the change in pH. I told you earlier that anything that has a pKa indicates it's a weak acid, and anything that's a weak acid can be a buffer. And so amino acids can be buffers as well, and they act as buffers. So this figure, or this, it's kind of a dumb scheme, but I'll show it to you. This dumb scheme can show us a little bit about what a titration plot looks like for the amino acids. Okay? If you look on the problem set videos that I work, I'll draw better ones than this because these tend to be a little odd. But here we can see, here is the titration plot for alanine. Alanine has no R group that can ionize, but it does have an alpha carboxyl and it has an alpha amine. Okay?
Is it a hydrophobic or a
Alanine is a hydrophobic because it has nothing that can interact with water. In the R group, has no side chain that can do that, okay? So the important thing are the ionizable groups, the alpha carboxyl and the alpha amine. We see the pH rising as we add, and here we're adding, by the way, NaOH. All right? We're adding NaOH to this solution. We're seeing the pH rise. Okay? The rising is going on, and we see it flatten. It's flattening right here. Why is it flattening there? Which R group? Oh, I'm sorry. Which group is being affected? The alpha carboxyl, because again, we're at about pKa of about 2.2, right? Within 1 pH unit of that, it's going to act like a buffer. We get out of that buffering region, and look what happens. The pH goes boing. The pH is rising rapidly, even though we've added very tiny amounts of sodium hydroxide. But then we get up to another region where there's buffering, and look what happens. Well, that's the alpha amine. And that's going to happen up around 9.5, thereabouts. Okay? Now, in that first figure that I showed you, there was a term that was on there that one of you mentioned that I didn't mention, but I'll mention it to you now. It's called a zwitterion. And I want to say a word about a zwitterion. A zwitterion is a molecule whose total charge is 0. Total charge is 0. Okay? Now, if its total charge is 0, that means it must have equal numbers of positive and negative charges, right? And we saw in that graph that I drew for you, or on that ionization earlier, that we had a molecule that had a charge of -1, 0, I'm sorry, of +1, 0, and -1, right? Let's go back to that since I'm referring to it here. So if we look at that ionization, okay, here's our molecule. Here's our amino acid. It's got a charge of +1. It loses a proton. It's got a charge of 0. It loses another proton. It's got a charge of -1. Okay? This guy's a zwitterion. Every amino acid can exist as a zwitterion. Now, let's think about these structures, and let's think about what did it take for this guy to become a zwitterion? What did it take? We had to pull that first proton off, right? We have a sort of a range over which we have a zwitterion, right? But in fact, when we look at a pH plot, what we see... Oh, don't start that again. Okay, blast, I shouldn't have gone away. What we see is there's actually, on the titration plot, there's a specific place where it will exist as a zwitterion, right? That first graph gives us an approximation. That rule I gave you about +1, -1 charge from the pKa, that is an approximation. That's all it is, right? The titration plot will allow us to see exactly where this is. Now, let's think about this. Here's a molecule down here. Let's say we're at pH 0. What's the status of the protons in this molecule? They're all on, right? We're just like that very first molecule we had before. We have a charge of, well, 0 from the carboxyl and +1 from the amino. So we have an overall charge of +1, right? We take that first proton off. Where's that going to happen on here? Where are we going to get that proton off? What's it going to take to get that proton off in terms of pH? We have to be more than 1 unit above the pKa. The place where I would wager 25% of the students on the exam will make a mistake on the exam is right here. They'll say, oh, there's that first proton off at the pKa. No, it's only half off there, right? To get it off, we have to get more than 1 pH unit above. We actually have to be exactly right there. Okay?
Wait, it shouldn't happen at 3.4 though? Or just
Hold on. Just bear with me, all right? It happens exactly right there, right? How do I know it's exactly right there? Remember I said this is an approximation when I say it's more than 1 unit above the proton is off? I said we can assume that. It's an approximation. The place, please, the place where a proton, where we have a zwitterion, is a precise place. There's a precise pH at which we have a zwitterion. That's known as the PI. The PI is the pH at which the charge of a molecule is exactly 0. The PI is the pH at which a molecule has a charge of exactly 0. It gets the approximation out of that thing that I gave you before. Well, how do I calculate this number right here? Right? Well, in this case, it's very simple. It's the pKas on either side of the place where it's 0. Well, there's only 2 pKas here. Right? If there's only 2 pKas, then it's the sum of this one plus this one divided by 2. The average of that will give me the pi. If this were 2.2 and this were 9.5, the correct answer on the exam would be 2.2 plus 9.5 divided by 2. I wouldn't even make you calculate that. 2.2 plus 9.5 divided by 2, you would have the pi of this amino acid. Now, what if I have 3 of these? Do I just average all 3? No. If you use the rule I just gave you, the 2 pKas on either side of the place where the charge is 0. Now, the TAs are going to be going through with you in class, in the recitations, pH plots for the amino acids. And you're going to see how you decide where the charge is here, where the charge is there, where the charge is. So you can find this magic place where the pi is, that is the 2 pKas on either side of it. Once you identify that, then you have the knowledge to calculate the pi. It's the average of those 2 pKas. Okay? So something that has, let's say, lysine, which has a positively charged R group, all right, it can, in fact, it'll have 3 places of flattening because it'll have 3 pKa values. So I've got to decide which are the 2 that are relevant. Does that make sense? Now, we'll see later in the term, actually in about next week, I think, where knowledge of pi gives us an incredibly powerful tool for understanding proteins. So this is not just an exercise in calculation, okay, but it's an important concept for understanding structure and function of proteins. Okay, questions about that? Yes?
Would the PI be like at the point of the question for that graph? Where
The pi would be right there.
is that, I mean, would the PI just approximately, if you had a graph and you needed a rough estimate of PI, would it always be a similar question?
It will. It will. Now, if you go and you look at these, that's why I don't like these graphs. So I'm going to show you, for example, their graph for aspartic acid. Okay, theirs doesn't draw this so clearly. Oh, sorry, I got to clear this and then draw it. Okay. In fact, it should be flat, up, flat, right? But here, because they're close together, it sort of runs them together, so I'm not real fond of their plots for this. But when you look at my videos online where I'm working these, or what the TAs are going to show, you're going to see some more defined flattenings where you can have no doubt that you're in a buffering region. Okay?
If you had like zoomed in, would that cause you to see it?
To some extent, but these 2 pKas are pretty close together, so it makes it a little bit more complicated. But you'll see the TAs will show you that a little bit more clearly. Arginine's a little better. Let me clear that one. Here's arginine. Okay. You can see the 3 here. Here's 1, 2, here's 3. Okay? Yes, sir?
So with the 3 pKas, which 2 pKas would you average?
You would have to calculate what the charge is at each place and assign that. So the TAs I'm not going to go through that here. The TAs are going to show you that in the recitations. Okay? But you understand the concept. You have to get the 2 pKas on either side of the place where it's 0. Okay? Okay. That's good. Let's see here. Let me get through here. So I haven't said much about primary structure, so I want to spend at least a couple minutes talking about that, and then we will, I think, finish with a song. Okay? Just something to keep you going. Okay. I've been doing all this talking about ionization, but I haven't said a word about primary structure. And that's how I started the lecture. Why did I do that? Well, we'll see that the charge of these amino acids affect the secondary, the tertiary, the quaternary structure of a protein. They affect all 3 of those structures. They don't affect the primary structure, however. The primary structure is essential, however, for all of the other structures of a protein. Underline that. The primary structure of a protein is essential for all the other. It determines what the secondary structure will be. It determines what the tertiary structure will be. It determines what the quaternary structure will be. The primary structure of proteins relates to the sequence of amino acids joined one to the other. Lysine, arginine, glutamic acid, valine, that's a sequence, one to the next. And the sequence happens because the amino acids are joined together by peptide bonds. You see a peptide bond being formed here. We see there's the peptide bond. It goes between the alpha carboxyl of one amino acid and the alpha amine of the next one. There's my R group at the first one. There's my R group at the second one. We'll say more about this next time. We see in this orientation that this guy has an N. This is known as the amino N because there's the alpha amino and it's not bound to anything. And there's the alpha carboxyl, there's a carboxyl N because there's the alpha carboxyl and it's not bound to anything. However, this alpha amine and this alpha carboxyl are tied up in a peptide bond. All proteins will have one free alpha amino and one free alpha carboxyl. All the other alpha amines and all the other alpha carboxyls will be joined in peptide bonds. Okay? So I can always tell which is the amino end of the protein and which is the carboxyl end of a protein. Okay, you guys have been patient. Let me see if I can get the audio going. I think instead of us, you can sing along, but I've actually got somebody who's going to sing for us. Okay? And I hope this works, so be patient with me. He's a way better singer than I am, so you may like that. And let's hear it. Sing along.
Lysine, arginine, axis, basic one you should not miss. Hallelujah, iron drift, fill the aromatic set. Rolling bends and cis has S. Lysine tar is the smallest. Then there's terp and tire and phe, structured aromatically. Acid and glucide chains of bond, say to protons au revoir. Glutamine, asparagine, bear carboxyl amines. 3 amino tiny snare, have hydroxyl groups.